In another segment, we looked at how Erasmus Darwin, Charles Darwin's grandfather, and a little bit later Wilhelm Preyer, spoke about the behavior of embryos, and it was sort of in the latter part of the 1800s when experimental embryology really started to gain some headway, because people were actually doing manipulations on developing embryos to see what goes on in normal development, but also some of the factors that could actually cause development to go awry. And then from that point on, more and more work was done in the 1920s. Xing Kuo did an awful lot of work on embryonic behavior, on the chick embryo in particular. And then again in the 1960s, more and more work started to be done. The research lab I was in at North Carolina was sort of one of the centers for doing behavioral embryological work. And so we're just going to look in this segment at some of the work that's been done in the area of behavioral embryology and a topic that is closely related to that. So we're going to start out by looking at how we can actually go about viewing embryos.
For biologists, chicken eggs offer a window into the process of development.
If you take a chick egg and cut a hole in the shell and throw it away, you can see the embryo floating on top of the yolk. It's right there, it's accessible. You can start to probe what's important for it to form by removing little bits and saying, does that disrupt the process? Or by moving tissue from one place to another and say, what does that do?
Cliff was following a long line of scientists using chickens to investigate how limbs develop. In the 1950s, John Saunders was one such scientist. Saunders and his team experimented on chick embryos just a few days old. They focused on little protrusions called limb buds, from which the wings would eventually emerge. In one experiment, Saunders took a small patch of cells from one side of a bud and transplanted it to the opposite side to see what would happen. When he came back a week later, much to his surprise, he found that the chick embryo had grown a second set of digits, one a mirror image of the other. That tiny patch of cells was clearly special. Somehow, it was telling the digits where to form.
So the moving around of embryonic parts like this is in fact something that began in the late 1800s, but as you can see, it's extended more to the, 1950s isn't the present time, but well into the 20th century. Okay, now in terms of embryonic development, what matters a lot is the kind of species that we're dealing with. Is it a precocial species or an altricial species? So what does all that mean? Well, Margaret Morse Nice in 1962 put forth a scheme, basically this continuum that extends from altriciality to precociality with many steps in between. And what she tried to do, and she was working exclusively with categorizing birds in her scheme, was to try to see where different species fell on the altricial precocial continuum. So she devised these different categories. Now what I'm going to do here is present her scheme. Again, she was trying to work only with birds. I'm going to try to fit in mammals wherever I can. It's not a perfect match, but basically some of it kind of fits. So we start with her category of altricial, and then her next step toward precociality is semi-altricial, and she defined two types of semi-altricial species. Then semi-precocial, so we're getting closer to the precocial end now. Precocial, outright precocial, but she also looked at 4 subtypes of precocial species. So let's look at these and just a couple examples of each one. Altricial. So all altricial species, according to Nice, the eyes are closed at the time of birth or hatching. There's little or no down feathers, or again, if we try to fit mammals into this scheme, little or no fur. Okay? They're unable to leave the nest at the time of birth or hatching, and therefore they're fed by their parents. Okay, so for example, This is an altricial species of bird, some species of songbird. You can see there's very little in the way of feathers, maybe just a few patches of down here or there, here and there. Not even much in the way of down feathers, just a little bit right when it hatches. Eyes obviously are closed, and it's not going anywhere. It's not for a little while. It has to stay in the nest, and it's going to be fed by its parents. So there's a fully altricial species right there, absolutely, totally dependent on parental care for survival. That is a zebrafinch. There's a European blackbird down below. If we try to fit some mammals in, so there's a squirrel in the upper right photo and a house mouse in the lower right photo. This is a highly altricial species. It's actually a highly altricial group of species. So if you're not sure what you're looking at, it happens to be a marsupial. And if you're thinking kangaroo, well, it's indistinguishable from a kangaroo, even though it's not. This is a North American opossum. And you can see how helpless it is at the time of birth. Basically, we have a remnant of what will become an eye. The mouth is pretty well developed. In fact, the whole olfactory area happens to be. The claws, very well developed, because they need to actually use these claws to climb from the mother's birth canal up into her pouch, where they will then undergo further development. But, you know, that the pouch is still not the embryonic period. It's when they are in the birth canal, coming out of the birth canal, where they transition from embryo to newborn, basically.
The opossums, like other marsupial mammals, the mother gives birth very early compared to what we would call placental mammals, things like us, like mice, like dogs. These guys, you know, they're a month and a half old, they're pretty big. When they're born, they crawl up here and attach to these nipples, and they're only a few millimeters in length at that stage. They are almost at an embryonic stage of development. About the equivalent of a human embryo at about 40 days of development. So they don't really have hind limbs, they have no hair, they can't hear at all at that point.
So this species doesn't have a pouch per se, but a whole bunch of nipples that are in this sort of indented area, for lack of a better term. I mean, you can see that, you know, the skin kind of opens up, but it's not an enclosed pouch like you would find in a kangaroo. But there they are, all attached to nipples where they have to undergo further development. But they've already been born. Okay, moving on to the continuum again, semi-altricial type 1. So you can see the defining characteristics. Now, both types of semi-altricial species, type 1 and type 2, have some down for mammals' fur. They're unable to leave the nest, though, therefore they're still fed by their parents. Type 1, the eyes are still closed like they were in the fully altricial form. So, examples of this: owls, there's some newborn owls. Doves, there's a squab, which would also include pigeons, of course, doves and pigeons. But you can see, compared with the fully altricial type, there's much more in the way of feathers here, and the eyes, of course, are closed. Here's a mammal I've tried to work into this scheme. Any idea what you're looking at? Okay, stop guessing. You won't get it. It's a panda. So a newborn panda might fit into this scheme. Now, for type 2, of course, they have all the top defining characteristics that you see in this slide. But now we're dealing with animals where the eyes are open at the time of birth or hatching. So that would apply to falcons, for example, and hawks, like we see here. Now, semi-precocial. So there is down or fur. The eyes are open. They stay at the nest even though they are able to walk away, but they don't. They stay at the nest. So therefore, they are fed by their parents because they're not walking away from the nest. Semi-precocial. So we have flamingos in the upper left. Penguins fit in this category. And gulls. There's our penguin chick. Yeah, I mean, they may walk like a little, a few feet at most away from the nest, but they don't go wandering off. The same with the flamingos and the gull chicks. So they are reliant on parents to be fed. Okay, now the precocial categories. 4 types. All types have down or fur, all have the eyes open, and they all leave the nest within 2 days of birth or hatching. Okay, so what defines type 1? Well, the type 1 precocial, they follow their parents away from the nest within 2 days, and they are still being fed by their parents. So the common gallinule, which you see in the upper left, the great crested grebe down below that, try to fit a mammal in there, perhaps an antelope would work well for this category. Type 2, they follow their parents, they're shown food by their parents, and they got to eat it themselves. So the parents do show them food, but they're not going to feed them directly. You can say, guys, there it is, go get it or starve. Bobwhite quail, well, all quail for that matter. And then in the middle, we have a red jungle fowl, and therefore domestic fowl fit in this category. Peacocks and other pheasant-type birds fall into this category as well. Now, the birds in this category, the parents have these food calls, and we looked at a food call of a jungle fowl and probably a domestic fowl. I don't remember if we did domestic fowls with food calls in a different segment. So they are actually sort of pecking at the food or semi-pecking. They might just point toward it with their bill in a downward motion while they utter these food calls when they're with their offspring. And then what you will see is the offspring will dash immediately to that spot and start pecking to get the food. And we see the same thing in ostriches and rheas. And this is a sandhill crane pointing downward. There it is. Go get it. Eat. Darn it. And it did. Now, type 3. They follow their parents. They find their own food. Parents aren't going to show them the food. Now, they got to find their own. So, the red phalarope, let's see, geese, ducks typically fit into this category. Finally, type 4. They are absolutely independent of their parents. These guys are on their own at the time of birth or hatching. And in most cases, the parents are nowhere to be found. So, here's a reptile. We can fit turtles into this category, where the eggs are laid in the beach under the sand, the parents are out of there, and then the turtles dig themselves out, like we see here. There's the head. Takes a while. I mean, my God, they're little turtle hatchlings. They're underground. And now they are off to the sea, hopefully, if they don't get disoriented. Sometimes that can happen, by the way, and they head in the wrong direction, and that's not a good thing. But more often than not, they'll just go right to the beach. Well, they're on the beach. They'll go right to the water. So that guy and his friends are doing the right thing. There he goes. Okay. Birds. This is an interesting species found in Australia. It's known by many terms: megapod, malleefowl, brush turkey, all the same thing. That is a hatchling. That is a mound that's actually made by the parents. They'll create it with sand. They'll add vegetation to it. They'll lay their eggs under the surface. So there is an adult, and it's created this mound. Okay, these can be pretty big sometimes, by the way. And notice that, there's different types of substance. This is sand up here. This is decaying vegetation. Basically, it's like compost. That generates the heat that incubates the eggs in this egg chamber that they've dug. And this down here is the main substrate of the earth. So they dug a nice little pit there where they have the compost, then the egg chamber, cover that with sand. The bird has temperature sensors, the adults do, in their bill, where they can add or remove sand if if the temperature sensors indicate there's either not enough heat being generated in this area of the egg chamber, or if there's too much, they can dig some sand out of there. Very interesting situation. But then as the chicks get closer to hatching, then they're not as concerned about temperature. Temperature is much more critical in the earlier stages of incubation as opposed to the very late stages of incubation. So then the parents are gone. They're generally nowhere to be found, and the chicks have to dig their way out. Now, for the photograph purposes, the sand above these eggs was removed, but here you can see a chick digging its way out. And there it is, it's out, and off it goes into the forest. It's totally on its own. Here's a very brief film that kind of describes this.
The male remains in complete control of the mound. He opens the mound for egg laying and checks the temperature. He alters its height according to the season to maintain the correct temperature in the egg chamber, and he continues his heavy work right through from August until March. Within a week of the last chick hatching, the male is again on the job, digging out the nest and preparing for the coming season. You'd think it'd be easier to make the female sit on the eggs and be done with it. After an incubation period of 8 weeks, the chicks hatch, about 3 feet under the soil. They dig vertically to the surface, moving up through the hot sand at a rate of about 1 foot an hour. On reaching the surface, they stagger to the nearest cover where they rest for an hour or so. Having rested, they move off into the scrub where they feed on insects and the seeds of acacia or small herbs. Each chick is completely independent and capable of flight on the day it hatched. Very little is known of their growth in the field, but it's believed they require 2 years to mature.
I don't know of any other bird species that's actually capable of flight at the time of hatching, or just a couple hours thereafter. So these birds are totally on their own. They are totally independent of parental care, kind of the height of precociality. Okay, so we've now seen the continuum of altricial to precocial. That feeds in to what I'm going to talk about now, which is the ontogeny of sensory systems, the development of sensory systems prenatally. And we're going to use as our example the precocial domestic chick embryo, which hatches on day 20. Now, I'm going to show you a film clip now that shows embryonic development. You'll notice that they say that it hatches on day 21. That's because the people that I work with count the first day as day 0, and these other folks count the first day as day 1. So that's why there's this 1 day difference, but it just depends on what your starting point is. So here we have 21 days, as I alerted you to. For us, that would be day 20. Doesn't matter all that much. It'll matter a little more in a moment when I show you something else. But you can see, this is all animation, of course, but it's done very, very well. Notice that big bump on the head? That will become the brain. That'll shrink down. The eye orbit, incredibly large at this point relative to the rest of the head. You can see the development of a beak, which actually starts out as what's called the egg tooth, which they use to cut the shell. That will actually kind of go away once they're born, once they hatch, rather. mammals are born, birds hatch. And you can see, relative to the size of the head, the eye has become a bit more proportional. Has a ways to go yet. The bump of what was the brain is now fully encased in the cranium. Of course, feathers are developing. Those limb buds have now pretty much developed into wings. And another thing I like about this is the correct positioning of the embryo that they've animated. This is that tucking stage, and then it's going to chip away at the egg, cut its way around, and hatch. And it's all wet and goopy because it's wet in there, but then it dries out probably within about 5 or 6 hours later. Okay, so that's what we're going to use as our model for looking at the development of sensory systems. So we're going to use our counting scheme, so day 0, not day 1. So 20 days to the point. They hatch on day 20. Okay. So on day 3, the first sensory system is actually fully developed, and it's a sensory system you've probably not heard of. It's called non-visual photic. Now, photic means light, sensitivity. Visual means vision, but it's non-visual light sensitivity, okay? The visual system has a long ways to go before it's going to become developed, as we'll see. So it's light sensitivity that the embryo exhibits as early as day 3. And these are some kind of receptors that are distributed in the skin. Very little is known about it. For one thing, it's very hard to study an embryo on day 3 of development. Really, really hard. We're not aware of the extent to which these sensors are found in other species. I know there is some evidence. I think it's in doves and pigeons. But what we do know is that it is developed in the chick embryo. And one way we know that, by the way, everything I'm telling you now comes from a variety of different pieces of evidence. It comes from behavior. It comes from neurophysiology and anatomy that converge to give us this information. We know, for example, that if you shine a light in the incubator on day 2, it has no effect on the hatching time of the bird. Starting on day 3, if you do it, and you just do it on day 3, that's what gets the birds to hatch about a day earlier. And I talked about that in another segment as the facilitation role of experience. So, day 3, this mysterious sense is developing. Okay, day 6.5, the sense of touch, tactile stimulation, is now fully developed. And in other words, if you touch the embryo at this point, it will twitch. If you do it before this point, it won't. So the tactile sense, fully developed on day 6.5. And by the way, this is where that whole brain area will become encased. That'll shrink down, and the brain will be encompassed in the skull. In fact, it's pretty much done on day 8. So yeah, 1.5 days later, the sense of balance, the vestibular system, is fully developed. If you turn the egg, you'll see the embryo tries to right itself in the opposite direction. And that only happens starting on day 8. Day 12, the auditory system, fully developed, and we're only 60% of the way through incubation. It can now hear sounds, and sounds do get through the egg. All the way to day 19, the visual system is fully developed. So notice we're leaving things out like olfaction because, as I've said in other segments, there's only a few species of birds where olfaction is a well-developed sense, and it's the chick embryo is not one of them. So, 1 day before hatching, the visual system is fully developed, and then, like I said, they hatch on day 20. Now, one of the reasons I'm showing you this is to talk a little bit about a phenomenon called homochrony. You'll remember in an earlier segment we talked about heterochrony, which is a change in the timing of developmental events across evolution. Okay, well, homochrony is actually a preservation in the timing of developmental events across evolution, because that's what we see with the development of sensory system across a very wide variety of animals, including mammals. Probably 30 some odd species have been studied embryologically in this regard. And these are the senses that actually show this phenomenon of homochrony. Tactile, the sense of touch; vestibular, balance; auditory, and visual. Across a very wide range of species, that is the order in which these systems become functional. So that is where homochrony is exhibited. A preservation in the timing of when these sensory systems become functional. That order is preserved. And of course, I've left out non-visual photic because we know so little about it. These are the ones, these sensory systems are the sensory systems in which we have the greatest amount of data from a variety of sources, again, neurophysiology, behavior, and so on and so forth. Okay. Now, the thing about this is in precocial species, and I'm not going to do all the subdivisions here that Margaret Morse Nice did, just general precociality, okay? All of these are developed before birth or hatching. They all develop prenatally. They are all fully functional before the animal is even born or hatched. Now, that is not to say they won't undergo further development and elaboration, because of course they will, but they are fully functional before birth. In altricial species, well, some of them are fully developed before birth or hatching. Others will develop after birth or hatching. Not too long after, by the way, but the animals will have become born or hatched before you'll see some of these, especially visual. That one is the one that's often delayed the most, not so much auditory. Okay, and it depends on how altricial the animal is in terms of what you're going to see here. But the point is, they're not all going to be developed prenatally as they are in precocial species. There is, however, one exception to this. There is one very altricial species in which all of these become functional prenatally, and that is us, humans. So we are the only altricial species in which all of these systems are fully developed before birth. And again, of course, they will undergo elaboration after birth. So that's just some of what's been done in behavioral embryology. There's been tons of more research done there, but that's all that I'm going to call to your attention in this course.